morning. It should be 4 o'clock in the morning there, right? Or maybe 5:30 now.
What city is it?
Texas.
Texas to be 1.5 hours. 5:30.
Right.
Which is not bad.
It's okay.
Yes.
Yeah, we can hear you. Can you hear us? Yeah, it is. You are audible now, but little more. Jatin, thoda और ज़्यादा.
Hello.
Yes.
What is having you?
Is it okay now?
Yeah, it's much better now.
Yeah, it is. Yeah, hello. I hope that you are hearing me. So I'm going to start my presentation. So first of all, I thank the organizer, Dr. Alok Bharti and Dr. Srivastava for giving me this opportunity to talk in this conference. I was supposed to be present there in person, but things changed and I was there. Hello. I hope that you are hearing me. So I'm going to start my presentation. So first of all, I thank the organizer, Dr. Alok Bharti and Dr. Srivastava for giving me this opportunity to talk in this conference. I was supposed to be present there in person, but things changed and I was there. I believe that he's also attending the conference. And also I did my postdoctoral training, one of my trainings with Dr. But I really appreciate the opportunity and I'm very thankful to both of them for providing me such a nice training that I was able to do something in my life. So thanks again. So today, like the previous two speakers, I'm also going to talk a little bit, not too much about the pharma nutraceuticals, it will be basic science talk. And the title of my talk is about the novel mechanisms which we found that regulate skeletal muscle mass. So my lab is focused mostly on the skeletal muscle biology with the muscle stem cells, muscle hypertrophy, atrophy, and also muscular dystrophy. So just to give you a brief introduction that the muscle mass is regulated by two processes. One is called myogenesis, which is the process of muscle formation where during the development and also after muscle injury, the progenitor cells, pluripotent stem cells, they basically commit to make myoblasts, and these myoblasts then differentiate into myocytes, and myocytes then differentiate into myotubes, and finally these myotubes mature to make the myofibers. These are the myofibers are basically the cells, muscle cells, which we all have in our body. And this process occurs both during the development as well as when there is an injury to the muscle, the whole process goes again. But once the muscles are formed, the myofibers are formed, they can undergo changes in their size. And you can see here that when in response to exercise, like we do all weightlifting and all those kind of exercise, or we take good nutrient food and growth factors, etc., they lead to muscle growth. There is no change in the muscle cell number, but there is also an increase in the protein content that leads to muscle hypertrophy. So if you look at the body of the weightlifter, it's not that they have more muscle, it's just that the muscle or the muscle cell has more mass. And that is because of the increase in the protein synthesis, in the protein content. Another mechanism by which the myoblasts or the myofibers can gain mass is through the fusion of these myoblasts, which are here. They can fuse directly to this and add like more protein or nuclei to the muscle cell. However, the muscle hypertrophy is good, but there are conditions where you can see that during aging and in some cancer patients and COPD, diabetes, there is a loss in muscle mass. So So many people you would see when they are sick, they lose their muscle mass drastically, and this process is called muscle atrophy. Muscle atrophy or wasting is very devastating in many disease conditions, and in cancer, for example, that 20 to 30% of cancer patients die not because of the actual cancer, but because of the loss of muscle mass. They are resistant, they are not able to tolerate chemotherapy and other interventions, and that is what leads to the mortality. The muscle catabolism or muscle wasting involves protein degradation, the inhibition in protein synthesis, there is evidence about oxidative stress, and there is also small amount of myonuclear apoptosis which goes on. So there are many pathways, signaling pathways that have been found to regulate muscle mass, and you can see this is a very busy slide. There is an IGF AKT signaling pathway, which is basically an anabolic pathway which leads to the protein synthesis and muscle hypertrophy, whereas the TGF-beta pathway is basically a negative regulator. It activates SMAD signaling, and the SMAD2 and 3 signaling basically causes muscle wasting. There is also the myostatin and activin signaling, then BMP signaling. All these, are basically play important role in the regulation of muscle mass. My lab is focused on understanding the role of these signaling molecules. Basically, this part of this thing, my lab identified 3 muscle wasting cytokines long back, but we also study these other molecules such as TRAF6, MyD88. We also look at the TAC1 and the NF-kappa B signaling pathways. But one of the things which is established in the muscle field is the NF-kappa B, and I got this training about NF-kappa B with Professor Bharat Agrawal, and I still run those MSARs which I learned in his lab. And thanks to that training, I think I made my career in this field. So the thing is that NF-kappa B is basically a muscle wasting transcription factor. It induces the transcription of several genes which causes the degradation of muscle protein. So when the people or even our lab identified that NF-kappa is an important regulator, I started looking at other molecules. We thought that if these NF-kappa, P38 and FOXO, all of them are causing muscle wasting, why not we look at the upstream signaling pathways, which might be like a better therapeutic target, because if you can inhibit one of them, you might be able to inhibit multiple pathways. So in that regard, we looked at some of the molecules, but today I'm going to talk about the TAC1. So what is TAC1? TAC1 is basically very important signaling kinase, and this is like in response to various cytokines such as TNF receptor, TNF interleukin 1 beta, T cell receptors. All they lead to the activation of the signaling complex here, which ultimately leads to the activation. These are ubiquitin ligases, TRAF2 and TRAF6, and once these ligases are activated, they ubiquitinate and phosphorylate TAC1 that leads to the activation of this complex, which is called the TAC1 signalosome. TAC1 then phosphorylates multiple downstream targets, including MAP kinases and IKK beta subunit of this complex, and this leads to the activation of NF-kappa. And TAC1 has also been found to activate SMAD2 and SMAD3 signaling pathway. And if you look at all of these, the AP1, NF-kappa, SMAD, they are all involved in muscle wasting. So we first started looking at the role of TRAF6, that what is the role of TRAF6 in muscle wasting, and that we were very happy and successful that if we blocked the TRAF6 in the skeletal muscle of the mice, we were able to inhibit muscle wasting in response to cancer, in response to denervation, and in response to basically starvation as well. But the next thing we started looking at the TAC1, we thought that if we can inhibit TRAF6 and we prevent muscle wasting, we would be able to do the same with the TAC1. But that was not the case. We got mice which are floss mice, like if you read, in these mice, this kinase domain is basically floss. So if you delete, cross the mice with 3, it deletes, it basically inactivates the TAC1. And we have this, initially we tried to use the normal 3 mice like we have they are deleted right from the birth, but we were unsuccessful. The mice died. They did not survive beyond 2 days after the birth. Then we had to use the inducible TAC1 inactivation strategy, in which we treated the mice with tamoxifen, and basically that leads to the inactivation of TAC1, and we started looking at the mice. So these mice survived, but we started the treatment at 6 weeks, which is basically 3 weeks, and we looked at the muscle, the weight of the mice at 6 weeks, and you can see that. If you delete TAC1, the weight of the mice is significantly, body weight of the mice was significantly reduced by the inactivation of TAC1 only in the skeletal muscle. And this is like, you can see here that in these mice, the TAC1 knockout mice develop kyphosis, which happens when there is a severe muscle wasting or there is a inhibition in the growth of the muscle. And we did some histological analysis with HNE staining. You can see these are basically myofibrous muscle cells. These are the normal mice, but if you delete the TAC1, there is a very drastic reduction in the cross-sectional area of the site of the blood. And this you can see here that by our strategy by treating the TAC1, this is the normal floss mice, but if you delete it, you can use if by toxic injection you can basically delete the TAC1 or truncate. And this is specific because TAC1 parasites and other proteins were not. So basically we thought that if we inhibit TAC1, we would basically prevent the muscle wasting, but it happened in the other way around, that TAC1 we found is very important for maintenance of or for the growth of muscle. We repeated these experiments in the adult mice where we used like almost 10-week-old mice and inactivated the TAC1, and we found the same thing. The mice, they basically lost their muscle mass, and there was like a drastic decrease in the muscle in the body weight. And histological analysis confirmed that it is because of the muscle wasting. You can see here the muscle.
I don't think that at all. Your forecast is 100%, 90% right. So it's in the window. It was like particular time. That would be great.
Can you hear me? Anyway, I'll continue. I'm not able to figure out whether I'm audible or not. So basically, you can see here that in the muscle there is muscle atrophy. You can see that the size of the myofiber is reduced even when you dedicate the TAC1 in the adult mice. So we looked at the mechanisms, and one of the important mechanisms we found that this is a sunset assay where you can see study the rate of protein synthesis, and you can see that TAC1 knockout mice in the naive condition there is a drastic reduction in the rate of protein synthesis. Also, the ubiquitin proteasome pathway is very important for muscle protein degradation during the atrophy condition, and we found that there is an activation of ubiquitin proteasome system. We also found that the autophagy was activated in these knockout mice, and also there was another signaling molecule which is called AMPK. It is phosphorylated, it is more activated, and it is known to cause muscle atrophy. So multiple pathways or multiple mechanisms are activated in these muscles after the addition of TAC1, which might be responsible for muscle wasting. Another very important observation was the mitochondria. We took out the muscle cells from there and we performed electron microscopy. So if you look at the normal mitochondria, into myofibril and subcellular mitochondria, there is like very good amount of mitochondria and very healthy mitochondria, all the cristae and everything are visible. But if you delete TAC1, we were surprised to find that these mitochondria were not healthy. They had this vacuum kind of structure, and there were like autophagosomes and all those kind of things were happening. You can see here at higher magnification the mitochondria was not healthy. We measured the stage 3 and stage 4 oxidation rate and the oxygen consumption rate, and we found that the knockout mice mitochondria are has very low oxygen consumption rate compared to the wild type. And we thought that all this thing is happening because the autophagy, which is an important mechanism to clear the dysfunctional mitochondria, we thought the autophagy is impaired, but when we took out the mitochondria and looked at the markers of mitophagy or autophagy, we found that there was no inhibition. If anything, there was an increase in the autophagy in the mitochondria of the knockout mice. I'm not showing the data here, but we found that there is, when you delete the TAC1, there is a disruption in redox homeostasis, and if you treat these mice with antioxidants such as Strolox, there is some improvement in the mitochondria function, and there is a rescue in muscle atrophy. So another experiment we found is that TAC1 is important when it is basically in the normal condition, but muscle growth, if you do exercise and all those kind of things, you see that there is a growth in the muscle mass. So we started looking at what happens to TAC1 when you subject the mice to the model of overload-induced hypertrophy. That means that there is sort of a resistance training kind of thing, weightlifting kind of thing. So we found surprisingly that TAC1 is highly activated in response to this overload on the muscle. But when we subjected these type 1 flox and knockout mice to the overload, you can see here in the normal mice there was a huge increase in the myofiber size, which is basically indication of muscle hypertrophy. But that kind of thing did not happen in the type 1 knockout mice. So this is the quantification which we showed that these data kind of shows that the TAC1 is very important in the muscle for its growth. And if you remove it, whatever like exercise, whatever training you do, there is no increase in the muscle mass. So, this leads to a very important question that if the TAC1 is sufficient, is important for muscle growth, if you, let's say, force activate TAC1, can you basically induce hypertrophy? You don't do any exercise, you just put the TAC1 back in the muscle and see what happens to the muscle. So this was the recent work we did, and what we did basically, we used this adeno-associated viruses, and you can see that the TAC1, it makes a complex with TAB1, and if you put only TAB1 or the TAC1 wild type, there is no activation of TAC1, but if you put this together, TAC1 and TAB1, and there is an activation of the TAC1. So basically, we co-injected these adenoviruses alone and in the combination, and you can see here, this is the quantification, that the size of the myofiber is significantly higher when you basically injected the muscle with the TAC1 and TAB1. And this is basically the Western blot data showing the phosphorylation of TAC1 at this critical, the kinase domain of threonine 184 and 187 happens only when there is a TAC1 and TAB1 were administered together. So these are the known facts, but we confirmed it that we need to put both of them together to activate TAC1. So we then started looking at some mechanisms. So one of the things which I showed previously is that if you delete the TAC1, there is an inhibition in protein synthesis. And when we found the opposite, like when we activated TAC1 through administration of TAC1 and TAB1 adeno-associated virus, you can see that the rate of protein synthesis was significantly increased. So we also then started looking at the signaling pathways, and the TAC1 regulates many pathways, including the NF-kappa P65, URF1, URF2, and P38 MAP kinase, and many other pathways which are maybe not directly regulated by TAC1, but we looked at the phosphorylation of all these molecules, and what we found is that P65 is activated by the co-administration of TAC1, TAB1, URF1, URF2, P38, and SMAD1 and SMAD2. SMAD2 are also basically activated. These pathways are all muscle wasting pathways. They call it muscle wasting, but it was surprising for us that we did not find muscle wasting. In fact, we found hypertrophy, but still these pathways were activated. And these two pathways, mTOR and AKT, they are anabolic pathways, and basically they usually increase the protein synthesis, but we did not find any difference in the activation of or the phosphorylation of AKT and mTOR. We then started looking at other proteins which are basically involved in the translation and protein translation, and you can see that these are the factors which basically initiate the translation. And surprisingly, we found that phosphorylation of EIF4B, and PRPS6, MNK1, and P90R6 kinase, all of them were significantly increased after activation of TAC1 in the skeletal muscle. So another protein which basically inhibits protein synthesis, PIF2 alpha, this is basically an ER stress marker, and it inhibits protein synthesis. We found that its phosphorylation was removed. So overall, we found that there is basically an increase in the activation of protein translation machinery in the skeletal muscle after activation of TAC1, and that is independent of mTOR and AKT pathway, which is classically known in the muscle field that this pathway is basically important for muscle hypertrophy. We did some cell culture experiments. We basically knocked down, so this is the muscle cells, they are not transfectable easily, so we have to make adenoviruses to knock down this thing. So we overexpress shRNA for TAC1, and you can see here that within 48 hours after putting the TAC1 knockdown of TAC1, there is a decrease in the size of these myotubes, which we call here. And the same thing we found is that there is a decrease in the protein synthesis. So then we did the reverse thing. We now use the myotubes, took them, and we overexpressed TAC1 and TAB1 to activate the TAC1. And surprisingly and consistent with our in vivo results, we found that the rate of protein synthesis was increased in the culture myotubes as well by the post-activation of TAC1 and TAB1. We looked at the various pathways which we found in the in vivo, and we found that the same pathway like the EIF, 4E, and this translation initiation factors, MNK, and ART1, they are all activated by in the culture molecules as well. Whereas there was not much difference on the, there was slight increase in the total mTOR, but there was not much increase in the phospho-mTOR. So, based on these findings, we thought that the TAC1 is a novel regulator of muscle mass, and it can induce this muscle hypertrophy independent of the AKT and mTOR pathway. And to confirm this, we basically inhibited this, mTOR signaling by using this compound called rapamycin. You can see that the protein synthesis is inhibited by treatment of the culture myocytes with rapamycin, but when we overexpress TAC1, TAB1, you can see that it is rescuing the protein synthesis, even in the condition when the mTOR are inhibited. So based on all these things, we felt and we discovered, and all the data suggested that in response to anabolic stimuli, there is activation of TAC1, and this TAC1 leads to activation of P38, and ART1 that leads to the activation of MNK1, and MNK1 is known to phosphorylate the EIF4 complex, which is basically the translation initiation complex. This complex then starts the protein synthesis. So another important aspects of TAC1 which we found was that it's role in the maintenance of neuromuscular junction. So what we looked at is that as you know that if you delete the TAC1, this is what we get here. But we look at the neuromuscular junctions morphology, and you can see here this is like the different staining, but these are our neuromuscular junctions. These are in the control mice, and these are basically in the knockout mice. So these things are disrupted here, and that happens during aging, and even in the cancer, during the cancer cachexia, it is known that the disruption of neuromuscular junction basically leads to muscle wasting. So we find this kind of phenomena in the naive condition in the TAC1 knockout mice. And these are some of the markers which suggest that there is basically a disruption of neuromuscular junction. So we've also investigated some mechanisms, and there are two, the pathways, which are important in the field of in the SMAD signaling pathway, which are basically important for regulation of muscle mass and maintenance of the neuromuscular junction. So the one pathway which is basically activated by GDF8, activin, and TGF-beta, this activates the SMAD signaling, SMAD2/3, and SMAD2/3 basically induces the expression of various components of protein degradation machinery. Whereas another pathway of this is activated by BMP signaling, BMP molecules, and this pathway is anabolic, and it leads to the activation of SMAD158, and this basically goes to the nucleus, and it induces the protein synthesis through the activation of AKT mTOR pathway. And there is a reciprocal regulation in these pathways. This is because of the competition for this SMAD4 molecule. Like if this pathway is activated, it inhibits the other one. So this kind of thing is happening. So we thought that we should regulate how the TAC1 is affecting these pathways. So when we deleted the TAC1 in the muscle, we found that both the pathways are activated. Both the pathways are disrupted. You can see the phosphorylation of SMAD158 as well as phosphorylation of SMAD2 is significantly increased in the TAC1 knockout. We basically performed some experiment to study whether there is some interaction between the TAC1 and the SMAD pathways, and you can see here IP by SMAD1 and Western blot by TAC1, and you can see that TAC1 is enriched in the denervated muscle. So basically during the denervation, it seems the TAC1 binds with the SMAD1, and you can see here the opposite reaction where we did the immunoprecipitation with TAC1 and immunoglobulin with SMAD1. And when we basically, performed this experiment where we basically used the TAC1 flox and denervated and the knockout mice and denervated the muscle, we found that there was more activation of the catabolic pathways of the muscle wasting signaling in the knockout mice. So it seems that when TAC1 is not there, TAC1 role is to protect the muscle from undergoing denervation-induced muscle wasting, and when the TAC1 is not there, there is more muscle wasting happening. This is just a quantification. We also performed some in vitro experiment where we used this pharmacological inhibitor of TAC1 5G oxygenol, and you can see that in response to recombinant BMP7 and 13 pathway, there is an activation of SMAD158. But this is inhibited when you add the TAC1 inhibitor. Same thing was observed when we knocked down TAC1 using the shRNA approach. There was a good activation of TAC1 SMAD by treatment with BMP 7 and 13, but this was inhibited by the knockdown of TAC1. And this plot here shows that we had a very good knockdown there. So overall, from this data, we showed that the TAC1 is very important for the activation of BMP signaling, which is anabolic signaling, which is important for muscle hypertrophy. And in one experiment, we basically put this TAC1 and adenoviruses, TAC1 and TAP1 adenovirus, and we performed the denervation in the mice. And you can see that the denervation, there is a strong decrease in the muscle cross-sectional area, but that was rescued by the force activation of TAC1. So overall, we found that the TAC1 is protective for the muscle, and if you basically put TAC1 in the normal muscle, it can prevent the muscle wasting. So I just a little bit 3-4 slides with the muscular dystrophy, and I think Nagendra had already showed all the slides to explain what is muscular dystrophy. But the muscular dystrophy again is a genetic muscle disorder, and it can be it happens because of the mutation in one of these cytoskeletal and the membrane protein, circulating protein. And Duchenne muscular dystrophy happens because of the mutation in the dystrophin gene. If there is a complete absence of dystrophin, it leads to Duchenne muscular dystrophy, where if the truncated form of the DMD dystrophin is available, it leads to Baker muscular dystrophy. So we are studying muscular dystrophy for past several years. So we just wanted to know what is happening to this is just to show that what happens in the TAC1, basically the muscles, the Duchenne muscular dystrophy muscles are like this is normal, and this is with the Duchenne muscular dystrophy. And the patient died within, in their mid-20s because they have this mutation in the dystrophin gene. There is a mouse model called MDX mice. It lacks dystrophin, and it shows kind of similar features as human DMD patients show. So we use this MDX mouse quite often to study the role of any signaling protein in the disease progression. So this is what happened in the MDX mice. The normal mice, at 2 weeks of age, the mice looks normal, their muscle looks normal, but around 2.5 weeks, the muscle necrosis starts. There is like degeneration of or the death of the muscle fiber, and then the infiltration of this inflammatory immune cells, and it continues. But in MDX mice, after 8 weeks, this muscle degeneration stops, and muscle more or less kind of recover, not completely, but that's the main difference between the DMD patients and in the MDX mice, that these show initial signs of necrosis, initial feature of muscle wasting, as happens in DMD, but later on the mice are fine and they live like almost 2 years, the normal lifespan. So, but what we found is that the TAC1 was activated in the skeletal muscle of the MDX mice, and you can see here the phosphorylation TAC1 and total TAC1. It happened only at the 4th week, but at the later stage there was not much difference in the level of phosphorylation for the total TAC1 level. So we wanted to know what is the role of TAC1 in the dystrophic phenotype. So we crossed the TAC1 muscle knockout with MDX mice, and we generated the control mice and MDX mice, treated these mice with the tamoxifen to induce the deletion of TAC1, and the mice were euthanized at 5 weeks. The inactivation started at the age of 4 weeks, and the mice were analyzed at 5 and 8 weeks. And you can see here at the 8 weeks, this is the normal MDX mice, but when we deleted the TAC1, we got the similar kind of thing, very small mice, and its quantification showed that there is very significant reduction in the body weight as well as in the muscle strength. So, we started looking at the histology, what happens to the mice, and this is the normal histology. You can see diaphragm, tibial anterior, soleus muscle, the muscle degeneration is happening, and all these kind of things. Surprisingly, we found that when we deleted the TAC1, many of these features were significantly reduced. You can see here that the knockout mice have like pretty good muscle, and there is not like much cellular infiltrate. We did the quantification both at the 5 weeks and at the 8 weeks. What we found is that really the various parameters of muscle injury, such as central repeated fibers and etc., were significantly reduced in the knockout mice. But the important thing was that although there is no pathology here or reduced pathology, but the size of the myofibers, if you look at this myofiber size, this was significantly reduced in the knockout, and this is the quantification. So what we basically found that we basically found that, the temporal regulation of the TAC1 is important. So if you delete the TAC1 basically at 4 weeks, there is an improvement in muscle pathology. Then we started looking at what happens if we, induce this, inactivate TAC1 at the later stages of muscle pathology at 12 weeks, for example, here. And you can see here that after the deletion of TAC1, there was no increase in the muscle pathology or anything. The muscles were normal, but only thing we found was that there was a significant decrease in the size of the muscle. And then, Anirban performed another experiment where basically he, at the age of 12 weeks, he put the TAC1 TAC1 together to activate the TAC1 and then analyzed the muscle at the age of 16 weeks. And as you can see here that, this is the normal control mice, but if you look at the one where the TAC1 is activated, the size of these myofiber is significantly bigger. So it seems like that if you put TAC1 to later stage, it basically does not do anything bad to the muscle. Indeed, it basically induces the growth of the muscle. And he looked at the signaling pathways also, and the same thing which we found in the normal mice, we also found that the pathways which are involved in the protein synthesis, they are increased after the activation of TAC1 in the dystrophic muscle. So overall, the research on the TAC1 in our lab has shown that the TAC1 is required for the growth and maintenance of skeletal muscle mass, and this is essential for maintaining healthy mitochondria. It causes, if you delete TAC1, it causes oxidative stress. TAC1 is important for the overload or basically weight-induced muscle hypertrophy, and supraphysiological activation of TAC1 is sufficient to protein synthesis and muscle growth. Furthermore, TAC1 is required for maintaining neuromuscular junction integrity, and it regulates BMP SMAD158 signaling, and also it prevents denervation-induced muscle atrophy, as well as it can, if you basically regulates, activate the TAC1 at an appropriate time point, we can also reduce the pathogenesis of muscular dystrophy. So with that, I finished my talk. I wanted to thank my lab members. So most of this work was done by Sajida Hindi, Shuichi Sato, and Kyle Anirban. Anirban is basically now a research assistant professor in my lab. He's running this project. And many other lab members have contributed. I have some collaborators, Dr. Brad Hill, he was at the University of Louisville. Shizuka Uchida, he's also at the University of Houston, and Shizuka Akira, he's the one who made this TAC1 floss mice and provided us. I also thank NIH for all the support they've been providing us for the past 15 years or so. Thank you so much. I don't know if I'm audible or not, but I'll be happy to take any question you have.
Thank you. Yes, sure. I don't know whether, can you hear us? Sure.
Yes, I can hear.
So
Yes.
We have a couple, we are running out of time, but a couple of brief questions for you.
Sure.
Yeah. Dr. Asur, can you hear?
Yes.
How specific TAC1 in muscular dystrophy? Is TAC1 plays any role in other bone disorders where that BMP also plays important role?
Yeah, TAC1 is not plays dual role. Basically, if let's say if you have like a TNF and all those inflammatory cytokines, they need TAC1 for their activation, and also they causes bone resorption. But in muscular dystrophy, we find that actually, if you look at the initial data where the inflammation play a role, that at that time point, if you inhibit TAC1, there is like a protection from the pathology. There is less muscle degeneration happening. So TAC1 has basically, that's what I said, that TAC1 is dual role. Like initially, it basically doing the bad thing to the muscle, but later stages when TAC1 is not activated, we put the TAC1 at that time point, it induces the muscle. So TAC1 has multiple roles. The knockout mice are embryonically lethal, so that's why it is. We use the floss mice to delete TAC1 in any specific cellular tissue.
Sure. Wonderful presentation. I have a quick question for you. Does IL-6 activate TAC1? Is TAC1 required for IL-6 induced muscular loss?
So interleukin 6 is not, the TAC1 is not involved in interleukin 6 signaling. It is involved in interleukin 1 beta signaling, as well as the toll-like receptor signaling.
Okay, thank you. Thank you so much. Wonderful presentation.
Thank you everyone. Thanks for the invitation again, and I appreciate this opportunity.